So welcome back, my dear students. I know you are very much, or not very much, a little bit worried about this crisis, this situation. But be bold and be positive. We shall definitely overcome this situation. But we don't have any alternative way. We need to continue the teaching learning process as the academic sessions are actually going on. So here in this video lecture tutorial, we are going to discuss actually the RNA processing and its type, its function. It's a very important biomolecule. You know that since the central dogma of molecular biology, you know the biomolecules are DNA, RNA, and proteins. The three biomolecules are very much important. Without synthesis or replications or transcription or translation or um, post translational modifications for protein synthesis, you know that in eukaryotes and prokaryotes, these are very essential things. So without prior knowledge about the RNA and RNA types and how they work, you cannot understand. You are not able to understand the detailed mechanism, how life is actually going on, and the cellular mechanisms are also going on. So, like the DNA is actually, you know, that there are several forms of DNA based on the structure and function. You know that A DNA, B DNA, Z DNA, and various types of DNA. Okay, and based on their size and linearity, they are circular, linear. So these are the types. So likewise, in RNA also, there are several types of RNA like mRNA, tRNA. Okay, so siRNA. So we'll accordingly discuss about here. You can see in the picture these are the biomolecules and different structure of the RNA. So RNA processing, how RNA process followed by the protein synthesis. Okay, so RNA processing in prokaryotes and eukaryotes. So basically, here is the some introductory part. You know that the best of the things are very known to you. RNA is much more abundant than DNA. That is a very important thing. So note it down. So in a cell or in any kind of organism, but there are some exceptions. Unicellular organism, for unicellular particular organisms, there are some exceptions. But normally, generally, the eukaryotes and prokaryotes, if you say, you know that there are RNA is the amount of RNA is more abundant than DNA. So why this is actually because of the production of the necessity of the production of protein. So once the necessity of production of protein is very essential, so you need to synthesize the RNA molecule abundantly than the DNA. So once DNA to DNA can be replicated via replication process, you know that. But in case of RNA, it has to be transcribed from the DNA. So RNA has to be need to be abundant in the cell so that it can follow the translation process by amino acid synthesis. Okay, so because your protein is a highly essential biomolecules is needed for your growth and development. There are several important differences between RNA and DNA. Is you know that is the ribonucleic acid and deoxyribonucleic acid is the basic difference. Here is only the ribose sugar. Here is the deoxyribose sugar. The pentose sugar in RNA is ribose and DNA is the deoxyribose. In RNA, uracil replaces the bases thiamine. U pairs with A. So you know that is a very known thing. RNA is single stranded while DNA is double stranded. So these are the basic things. Okay. So now is the important thing is that RNA. Why we are discussing about the RNA structure and RNA processing, we need to know about the structure. The structure actually is not an easy process. If you go through any kind of biomolecules, like the protein, like the DNA, and like any kind of RNA or any carbohydrate, glucose molecules, or anything else, the biomolecules in a living system. Their structure is actually having the primary structure, preliminary structure, or primary structure, followed by secondary and tertiary structure. So primary structure means in depth, inside the molecular level, atomic level, or chemical composition level. This is the primary structure. You can understand. You can see here. So it's very difficult to remember for your exam, but here you can see only the primary structure indicates how adenine, cytosine, guanine, and uracil here, not the thiamine, uracil, they attach. Here is the ribose nucleic acid, chain-wise ribose nucleic acid. This one here is also phosphodiester bond. Okay, and it makes the phosphate backbone here. It makes the phosphate backbone. This is the free 5 prime. This is the 3 prime end, and here it attached with the guanine, uracil, cytosine, adenine, and that side also similarly the phosphate sugar will be there. Okay, so this is very complex and detailed primary structure. Just overall thing is that you need to remember only the uracil is present in case of thiamine. Okay, next is the secondary structure. So primary structure is already over. Primary structure means the detail atomic level and molecular um, uh, elemental level, actually the study. Now the secondary structure means a little bit, if you zoom it out. So already you zoom in is in depth in the primary structure. Now you zoom out a little bit, zoom out. Okay, so then you can see the secondary structure is a little bit bigger form than the primary structure. So here this is the actual structure of the RNA molecule. You see, it's like the handle and loop, again loop, loop, loop. There are hairpin like structure. So these are the common term, are very much important for your MCQs and for your future references also. So this is very important, and if you want to note it down, you can pause the slide and you can write it down. Single stranded region formed by unpaired nucleotide. Single stranded region formed by unpaired nucleotide. Unpaired nucleotide. These are not paired nucleotide. You see here, these nucleotides pair are not based on their adenine, purine, and pyrimidine bases. Okay, is the unpaired. Duplex means double helical RNA, A form with 11 base per turn. So 11 bases per turn means you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, so this is one loop, this is a major loop, major loop. So in between 11 bases per turn, one turn, you can, okay, 3, 4, 5, 6, 7, 8, 9. Okay, so now see, next is the thing is hairpin. Hairpin is the duplex bridged by the loop unpaired nucleotide. Okay, so duplex bridge here, you can see the duplex bridge here. See, okay. So it's already mentioned single stranded region. You can see here single stranded region, and duplex means the B duplex here is making duplex. Hairpin is the hairpin. You know, the girls maybe better knows that the hairpin means how they use the hairpin. Okay, so this is the upper top portion. There are some bases, open bases is the hairpin. Internal loop in the D, internal loop in between is the major minor loop, internal loop. It is a nucleotide not forming Watson Crick base pair. So here you can see this is not helical and Watson Crick model. It does not follow the Watson Crick model. See, here is the open base, open base, open base. So there are some open bases. D, internal loop. Now E is the bulk loop. Bulk loop is here. See, is like the budding. Okay. So if you can remember the bud or budding or like a tumor form. Okay, so this is the bulk loop, is called the bulk loop. Junction F is the junction, means suppose if you consider the railway junction, so from this junction several lines are going on towards different places. So this is the junction when the hairpin, internal loop, or even bulk loop, all they get attached here. This is called the junction position. Okay, and pseudo knot is means the hidden knot. Pseudo means, the hidden knot. Pseudo knot and hidden knot is a similar thing, means tertiary interaction between the bases of hairpin loop or outside bases. So sometimes if this is breaks, then it will be open, and here is also a pseudo knot. This is also called a pseudo knot. This is also pseudo knot, means once this one is breaks, so it will be break, like it will make a single stranded region. And this one, if this one is breaks down, though this will make a single strand, like the a single stranded region, this one. Okay, so that's why this is called a pseudo loop. Okay, so I hope you understood this slide. So now is the primary is over, secondary is over, now is the tertiary structure. Tertiary structure means now you zoom out a little bit bigger form that can be seen under the microscope. So here is the covalent and secondary tertiary non-covalent hydrogen bond. This is the actually nucleotide sequences which actually forms the amino acid structure, and amino acid structure when the amino acid chain is synthesized. This is the primary, the primary means I have shown you. Primary means at the deeper level, base level. Secondary means is a little bit zoom out, is a bigger form. And tertiary means more bigger form that can may be visible under the microscope. So this structure is scanning electrons microscopic structure. So this is the actually tertiary structure of protein molecule. Okay, an RNA molecule and protein molecule. So if you go the quaternary structure, there we will be able to see the actual number and actual form. So here, RNA structure is the overall thing, is also called primary, secondary, and tertiary. Is the 1 degree, 2 degree, and 3 degree. Primary is the 1 degree, 2 degree is the secondary, and third is the tertiary. You see, this is the linear, a single stranded form is the primary, and here is the structures like a loop, hairpin, and pseudonodes, bulb. All these things will form at the 2 degree, and 3 degree means tertiary structure. You can see the a complete substances or complete a material. Okay, so this is the